Hi class, welcome to lecture number 10. This is the first lecture in Unit 4. So we're making a pretty big transition here in Unit 4. If you think about what we've done so far in the course, our first lecture, or our first unit I should say, was really an overview of the microbial world, and we talked about the general characteristics that most microbes share and how they got to be here, how they evolved on this planet, and what their major commonalities are. In the second unit, we talked about how microbes obtain and process energy. We talked about their metabolism and their metabolic processes. In the third unit, we talked about the genetic expression of prokaryotes and how that differs from eukaryotes. We talked about how genetic variation can arise and how gene expression is controlled in most basically in bacteria. And so this fourth unit is really a very abrupt turn or abrupt change in how we've been dealing with the course so far, because now we're actually talking about us. We want to understand, ultimately what we're trying to get at is how to understand or how to describe our what's called the host microbe relationship. In other words, how do we as humans interact with microbes in both homeostasis and health and disease. And in order to understand that, we first have to understand our immune system. So for most microbiology courses, especially the medically oriented microbiology courses, we have to spend a few units on immunology. And immunology is actually a major course of my study in my grad school, and it's a really fun topic because it's so incredibly complex. But it's also a very frustrating topic because it's so incredibly complex. So don't get frustrated with all the details. I try to paint as broad of brushstrokes as I can in terms of the signaling pathways and all the little details that are involved in our immune system, because it's crazy. It's so complicated because we want that redundancy. If you think about how you want a system, a really important system that can't screw up and that can't fail, and has to be effective, you want a lot of redundancy built into that system. You want a lot of checks and balances, and you want a lot of crosstalk. So our immune system is really, really complex. So I try to simplify it as much as possible for you, but understand that it's a very complex topic. So let's start. Also, I guess kind of an abrupt change in my beginning slides too. I think I usually have just a plain white beginning slide for each lecture, but I love this picture because this is actually one of the major cells of our innate immune system. This is actually a macrophage here, engulfing some bacteria. It looks like maybe some type of diplobacilli, right? Some paired up bacillus bacteria, and macrophages eat bacteria. They're a phagocyte, and so a phagocyte basically means a cell that can eat other cells. So here we see a macrophage engulfing and digesting these little pink bacteria. So let's go ahead and review just really, obviously I hope you've already went over the learning objectives for this unit. You can see there's about, it looks like I crossed out a couple of them. There's about 11 or 12 of them. And we really are going over the first 2/3 maybe of the chapter 14 of the Nestor text. So before we talk about the innate immune system, so lecture number 10 is all about innate immunity. This is lecture 10, whereas lecture 11 is all about adaptive immunity. But let's talk, before we start talking about innate immunity, let's talk about what they have in common and really what the general purpose of our immune system is. Ultimately, the end purpose of our immune system is very simple, and that's simply to keep us alive. To basically, a fancy way of saying that would be to maintain homeostasis. So really what our immune system is doing is keeping us alive against a constant barrage of attacks from the external environment. So our immune system has a really, really tough job. It has to be constantly vigilant because bacteria are there's an unending supply of them. And then don't forget that we learned bacteria can double every 20 minutes. So if you've ever been around a decomposing body or decomposing anything, bacteria take over that organic material in a day. Right? And so imagine if our immune system ceased to function even for a few hours, we would be dead in the water. That's a bad analogy. We'd be in big trouble, right? We would be in really bad shape because that bacteria has had all that time to basically get inroads into our body. So our immune system has to be always on. And it has to not only be always on, but it has to be ready against a huge variety of external forces. Think about it. It's not just bacteria, it's fungi, it is viruses, as we're learning during this pandemic, it's parasites, it's other small eukaryotic pathogens, for example. So yeast, believe it or not, you can die from a yeast infection. There's a type of yeast infection called thrush, and it's actually one of the most common ways that people with advanced AIDS can die. You can die from a yeast infection if your immune system is compromised. But at the same time, our immune system can't damage our own body, right? It can't cause excessive damage. So it has to be very specific and very directed against those external forces. It can't damage our own tissues and our own cells, and it can't target our normal bacteria. Because remember, we're covered in bacteria. Our external, everything that's external, and even a lot of our internal compartments have bacteria as part of the normal microbiota. And if we destroy that, we're destroying a really important part of our homeostasis. And that can lead to a state called dysbiosis, which basically means an imbalance of bacteria, which can actually hurt us, believe it or not. We need those bacteria. Now, let's talk a little bit about these two branches of immunity. Our immune system is broken into two major but overlapping spheres. And the first one is innate immunity. The second is adaptive immunity. Now, innate immunity, I love innate immunity. I think it's such a cool topic because it is the cells in the signaling pathway that make up our innate immune system are the very same things that are in the most simple organisms, like nematodes, like these really, really ancient organisms that have been around for hundreds and hundreds of millions of years. And it's a commonality throughout all of these higher level, in other words, eukaryotic life forms. And I think that's so fascinating to think about, that there's a big part of our immune system function that we have in common with all of these seemingly unrelated animals. And so that's yet another kind of indicator that we all arose from kind of a common ancestor. So this innate immune system, the characteristics of our immune system, our innate immune system rather, are present at birth. In other words, we are born with some forms of immunity, and they're encoded. So germline encoded basically means that it's part of our DNA. We don't acquire these as we kind of grow and survive and learn about the external environment. These are things that were given to us by our mother and our father in our genes. So germline is kind of another way of saying it's in our DNA. So what are we born with? Well, we're born with anatomical barriers. As you'll learn very quickly here, the best part of our innate immune system function is really our skin, is basically having healthy, intact, non-compromised physical barriers. So anatomical barriers are probably one of the biggest players in innate immunity. Another characteristic of innate immunity is that it's very fast acting. It's the first it's the first component of our immune system to act because they recognize patterns. So they're not going to be, our innate immune system is not very specific. It's going to recognize more or less all bacteria. They treat all bacteria as the same. And they usually respond, a big component of the response of innate immunity is through chemicals. And so either small proteins or other types of chemicals. And there's also a very large component of the innate immune system that is a physical response. For example, fever, inflammation, and so on. On the other hand, adaptive immunity has its own unique characteristics. And I'm just going to kind of briefly go over these because we'll talk about them in detail in the next lecture. But adaptive immunity is specific to higher order vertebrates, and it develops throughout our life as a result of encountering foreign material. So this is how we kind of gain immunity. This is an integral part of adaptive immunity, of course, or an integral application of adaptive immunity are vaccines. And we'll talk about vaccines in Unit 5 because vaccines literally are they're exposing our body to foreign material, foreign antigen, and it's teaching our body what the enemy is. So we've all heard about this talk with the pandemic and how it would be so great to get a vaccine, and there's different timelines on when we're going to have that vaccine come online. And so if we could teach our body how to recognize coronavirus right away and take care of it before it can make us sick, that's a huge advantage in the fight against this COVID-19 virus. So another kind of hallmark characteristic of adaptive immunity is that it learns and remembers. So innate immunity doesn't learn or remember anything. It just is, right? Our skin doesn't change. It doesn't adapt to any kind of foreign pathogen and change the way that it works based on that foreign pathogen. Our skin just is, right? It's part of our body and is present at birth and is encoded in our DNA and so on. Whereas there are certain cells in our adaptive immune system that will actually take an insult, like a foreign antigen stress, and take that stress, that foreign antigen, and kind of keep it in its back pocket. And these are called memory cells. And this is, again, a very important application of vaccination. So we actually have cells in our own immune system that can learn from an infection and remember what that infection looks like for the future. Unfortunately, this means that it's kind of slow acting. So we can't have many aspects of that adaptive immune response. Takes a few days to come online. And in the meantime, it's all that innate immunity that's kind of protecting us while our adaptive immune system gets ramped up if needed. And then the last characteristic of adaptive immunity, also really important, is that adaptive immunity is very, very specific. For example, all of us right now have antibodies in I shouldn't say antibodies. We have memory cells, B cells and T cells in our body that specifically recognize coronavirus. Believe it or not, you would be surprised to hear that, because coronavirus is a very broad category of viruses. There's lots of viruses that are classified as a coronavirus. But unfortunately, because those memory cells are so very specific, it's not our immune system isn't recognizing that specific COVID-19 antigen, and therefore can't protect us from that. So our B cells and our T cells that make up these antibodies that protect us are so highly specific that they only recognize very, very specific, they're called epitopes on viruses or bacteria and so on. But again, I just said I'd only briefly go over adaptive immunity, and I talked about it for like 3 minutes. So one good way to kind of think of adaptive versus innate immunity is that adaptive immunity are the first responders. They're the people that's going to be first on the crime scene, and they're going to respond, and they're going to clean up the scene, and they're going to contain it, and they're going to prevent the damage from spreading. Whereas adaptive immunity, they're kind of like the detectives. They're going to kind of get the phone call the next day and say, you need to investigate this and figure out what's going on and make sure, that it doesn't happen again. All right, so let's go ahead, and I need to pause it really quick. My phone keeps ringing. Might be important. Hold on. Okay, it was important. My friends, I have a very important category game over Zoom with my college friends. So we're supposed to play tonight, and we're making sure that we're all going to be online at the same time. Okay, so let's continue on with innate immunity. So to carry on, a lot of times when we talk about immunology, we make a lot of analogies to war or defenses or crime, because it's a battle, right? It's so if you think about, and it's not our immune system is really more nuanced than that, because our immune system, believe it or not, works in concert with a lot of bacteria. So it's called our microbiome. But let's think about really how our immune system interacts with pathogens, really. And it really kind of is a battle, and it's keeping that homeostasis, that balance of healthy function the same, right, throughout in our body. I totally lost track of what I was trying to say. So when we think about immunity in general, and in particular innate immunity, there often are a lot of analogies to war or to defenses or to crime, because it's easy to think about the way that our body and pathogens interact as like a battle. So if we think about our body like a castle or like a compound that we need to protect and defend, we can kind of think about innate immunity in 3 major branches. The first line of defense is going to be our security walls and basically our physical barriers. And these are things that are like that physically prevent microbes from entering. So think about our skin and our mucous membranes, and then any chemicals that are found on our skin or in our mucous membranes that prevent bacteria kind of from getting a toehold. Remember that humans have an external and internal environment, and bacteria in almost every, in fact, I can't think of an external environment that bacteria aren't in on our body. And that's fine, but what we have to do is prevent them from getting into that internal environment. The next line of defense is our sensor system. And so you can kind of think of those as our security cameras that are designed to kind of stroll the perimeter, right? They're walking along the top of the rampart on the castle, or they're walking along the border, the fence, or they're the guards up in the watchtower, making sure that no one's trying to climb over the fence and so on. Or I'm sorry, climb into the compound. And these sensor systems only turn on when the invader has gotten past our physical defenses. So for the most part, we call our sensor systems our sentinel cells. And these sentinel cells have what's called very specific receptors on their surface called pattern recognition receptors. And another very important part of our sensor system is our complement system. However, both of these really act over here as well. So our pattern recognition receptors and our complement systems not only sense that there has been a breach of our first line of defense, our physical barriers, but it's also it's first going to recognize and amplify that signal. So they're the ones that are going to let everyone else know that we have a pathogen that has gotten past our first line of defense. And they're also going to have some type of effector function too. So let's talk about our last branch of innate immunity, and that's the security teams, right? So if we have a sentinel, if we have a guard, and they're kind of watching the security system, and they see someone sneak in, they're going to call the alarm, right? They're going to turn on the siren, and they're going to put out an all call, let people know that the north wall has been breached, or whatever it might be, right? And then we're going to have effector teams come in and eliminate that threat. And we have lots and lots of effector functions in the innate immune system. And they're the ones that are going to be responsible for either directly killing that So let's just make it simple right now and talk about bacteria. Again, it could be a virus, it could be fungi, it could be lots of other things. But let's just imagine that we have a bacterial pathogen that has gotten past our first line of defense. We have a variety of responses that will either directly kill that bacteria or can phagocytize that bacteria or other cells that bacteria has invaded. So there's lots and lots of effector functions that we're going to go over at the end of this lecture. So again, just kind of keep this flow, this way of organizing the innate immune system in your mind as I go through this lecture. So let's talk about those security walls first, our first line of defense. And actually, before I even talk about first line of defenses and physical barriers, let's talk about natural resistance. And this is kind of, I almost think this is, I don't even think of this as our immune system. It's really just a function of who we are. And I know that you as students, and even I lose sight of this sometimes because we're a medically oriented microbiology class. But remember that there are lots of other, like there's environmental microbiology courses, there are industrial microbiology courses. So if you were to look at this planet as a whole, there are millions and millions and millions of bacteria and viruses and tiny little microbes that want nothing to do with us, right? They don't want to be in a human. They don't want to be in our gut. They don't want to be on our skin. We're not a hospitable host to the vast majority of bacteria and viruses and so on on this planet, because we have a very specific pH in different portions of our body. We have a very specific temperature, and so on. So imagine if you think, so I guess I just want to remind you that sliver of bacteria that are medically important to us as pathogens really only represent a very, very small fraction of all the microbes that are on this planet. So if you think about the bacteria that could be a pathogen, They're probably a mesophile, right? They essentially need to or want to grow at around 37 Celsius. They're probably a heterotroph. I mean, if they're an autotroph, they don't, they don't want to be on us or in us, right? They want to be where the sunshine is. They want to have access to that energy source. What else are they? They're probably, depending on where they are in our body, they're probably not very halotolerant. They can't put up with large amounts of salt. We do have a range of oxygen requirements. Of course, any bacteria that are in our external environment are probably either aerobic or facultatively aerobic. We do have some anaerobic environments in our body, like deep in our gum line, for example, there can be some anaerobic bacteria. Certain places in our gut can be highly anaerobic environments and so on. So only very specific bacteria really even want even really want to be on us. So we have a natural resistance to the vast majority of microbes that are on this planet. What about those bacteria that actually want to cause problems for us or will cause problems for us? How do we keep them from doing that? By far, our best defense against bacteria is our physical barrier. In other words, our skin and our mucous membranes. I know that many of you are CNAs or either training to be CNAs or work in a medical environment in some ways. This is why it's so incredibly, like in long-term care, in a nursing home or hospice care or rehabilitative care and so on, why it's so incredibly important to make the bed a certain way or turn the patient and roll them over and prevent bed sores or prevent any type of interruption of that physical barrier, because this is hands down the best way that we prevent bacterial invasion, is having healthy, intact, non-compromised skin and mucous membranes. Our skin is like, again, think about those big medieval castles, right? Imagine trying to scale this huge, tall, if you watched not Lord of the Rings, the other one, Game of Thrones, right? It's like trying to cross the wall. They built that huge wall that you just can't, it's too big to climb. It's slippery, you fall off of it. And that's really what our skin is like. Remember, our skin has, I'm going to skip ahead and then I'll come back to this slide really quick. Our skin, the epidermal layer of our skin, has all of these differentiated little epithelial cells. And by the time those cells get out to where we can see them, right? Any part of your skin that you can see and that you can look at is actually dead, right? Our skin cells differentiate to the point where they lose their nucleus and they become full of keratin and they die and they flake off all day, every day, right? Most kind of a gross thing to think about is that most of the dust that's in your home is really your dead skin. That's super gross to me, but it's true. And that actually is a huge benefit. It's a huge benefit to you because think about bacteria are trying to grab on, right? They're trying to hold on and they're trying to penetrate that skin. And if our skin cells are falling off, remember, our skin cells are way bigger than bacteria. So you have this teeny tiny little bacteria that's on one of our skin cells. And if our skin cell falls off, that bacteria is going to fall off right along with it. So big advantage to our skin is, one, we have this external dead layer that keeps falling off. And then we have that bottom, that basement membrane of connective tissue that is so tightly kind of connected to one another that even if our outer cells become compromised, that bacteria can't get past that those really tight connections that our basement membrane makes. Remember that our dermis, think about what leather is, right? Leather is usually cow, I think, or whatever kind of animal, but let's just say, it's made from a cow. It's the dermal membrane of an animal. And that tissue is so tightly held together that not even water can get through it, right? You can make a water pouch out of leather. That's what they did until, better, more technological solutions came along. So, when you think about how good of a barrier our skin is, it's pretty incredible. And again, this is something that all animals have. On the other hand, we have our mucous membranes. And remember that our mucous membranes are also considered external. If you talk to a physiologist, they'll say that a human is really just a complicated tube. I think that's kind of a funny way of thinking about it, but this is really kind of a schematic of what our body looks like. in a very simplified way. And the pink areas are all the areas that are still external, but for various reasons have to be covered in a mucosal membrane as opposed to that kind of dead epithelial layer of our skin. And our mucous membranes are bathed in mucus, right? All of these pink, if you think about where all these pink coverings are in our body, they all have some type of slippery coating to them. And that slippery coating is important, one, for function of the mucosal membrane itself, but it's also protecting us from those microbes. So it's it's basically creating a slime layer in which those microbes get trapped. And then there are mechanisms in our body that can move those microbes along in a certain direction. So for example, peristalsis moves anything that is in our digestive tract, upper digestive tract into our stomach. And then of course, once it's in our stomach's basically a big pit of acid that these bacteria are going to die in. And then alternately, it could move it up towards our, out of our lungs, up towards our sinuses so that we can sneeze it out or swallow it. So if you've ever taken, again, for those of you that have taken maybe anatomy or physiology, I'm not sure which one you talk about it, but we call it the mucociliary escalator. So again, let's kind of think about what our mucosal membrane looks like. We have all of these mucus producing cells. I think they're called goblet cells. But it's secreting, right, this big slime layer. And then right beneath that slime layer are all those little kind of hair-like protrusions on our membrane cells. And that's going to move that mucus in a very specific way. So we want the mucus to move away from our lungs. We do not want bacteria in our lungs. That's a big problem if we have bacteria in our lungs. And we want it to move up near our, what is it? Epiglottis? I forget. We want it to basically to move into our esophagus, right? We want all those bacteria in that mucus to move those bacteria away from our lungs and into our esophagus so that we can swallow it and it ends up in our stomach where the bacteria will be killed. In other words, our mucous membrane has systems for funneling those microbes out. And then don't forget that, of course, our cells are constantly turned over in our mucous membranes as well. So big, if you have to choose one type of immune branch of our immune system or one aspect of our immune system you don't want to lose, of course, it's our physical barriers. Now, not only does our skin and our mucosal linings have a good physical defense, they're coating themselves in chemicals, in antimicrobial substances. And one of the best ones is simply salt. Our skin is very salty. It comes from our sebum secretions and our sweat. And this is going to inhibit most bacteria. It's going to inhibit all bacteria except for bacteria that are either halotolerant or maybe even halophilic. Halophilic is pretty high. That's a pretty high salt concentration, but you have to be halotolerant in order to survive on our skin. And most bacteria are not halotolerant. They can't survive in such a dry, salty environment. One exception, of course, is a commensal bacteria that is on all of our skin. And we've actually looked at it quite a bit in lab, and it's called Staphylococcus epidermidis, right? It's found on the epidermal layer of our skin, and it is halotolerant. Another chemical, it's actually an enzyme that is secreted by our body. We've actually talked about it quite a bit already, is lysozyme. Remember that lysozyme is a cool little chemical or cool little enzyme that literally dissolves peptidoglycan. And if you break apart peptidoglycan, you're basically breaking apart the cell wall specifically of bacteria. Remember, peptidoglycan is only ever found in bacteria. So, So when our body secretes lysozyme, it's specifically targeting bacteria. And it's found in lots and lots of our secretions, tears, saliva, mucus. I think it's even in our sweat. And also some of our cells secrete it. We'll talk about that later when the cells engulf bacteria. They want to destroy those bacteria. They basically hit them with some lysozyme. We also secrete these enzymes called peroxidases. Peroxidases secrete hydrogen peroxide, and this directly kills many types of bacteria except for bacteria that contain the enzyme catalase. Remember that catalase can neutralize hydrogen peroxide. So again, this is in many of our secretions, saliva, breast milk, body tissues, and certain cells have peroxidases as well. The next two, lactoferrin and transferrin, they're molecules that bind to iron. And so it's called lactoferrin in our secretions and transferrin in our blood and tissues. And bacteria need iron. It's a really important cofactor for a lot of enzymes that bacteria use. And if they can't find iron, their enzymes aren't going to work. And if their enzymes don't work, they're going to die. So one of our body's kind of generic responses or generic ways to prevent bacteria overtaking us is to sequester any available iron and not make it available to bacteria. And then finally, I want to talk about some I don't want to talk too much about them because I don't really actually know much about defensins. Defensins are tiny little peptides. They're like very, very, very small proteins, very short stretches of proteins that seem to have some function in antibacterial responses. There's a lot of really interesting research being done about them. They were discovered not that long ago. And it's kind of a cool area of research right now, but I just wanted to mention them because it's kind of one of the newer fields for antimicrobial research right now, defensins. All right. So an often overlooked aspect of our first line of defenses is the bacteria that normally inhabit our body. Remember that we're covered in bacteria. We have bacteria everywhere, on us, in us, on any surface that's considered external. We have bacteria on our surface. And there's bacteria that are considered, we basically call them commensal. And that means that they live with us, they don't cause us problems, and they actually help us in many ways. And the big way that they help us is through competitive exclusion. In other words, they're making, how should I say? They're crowding out any potentially harmful bacteria. So imagine if there's a bacteria that we don't encounter very often and it wants to get on our skin. Well, it's not going to find a lot of space because our skin is already covered in bacteria. So that bacteria that's kind of already friendly with us is going to cover up any potential binding sites. It's going to gobble up any of that iron that might be laying around. And it also, don't forget that bacteria attack one another. So it's possible that the bacteria that is kind of resident bacteria on us can secrete chemicals or proteins or some type of factor that's not harmful to us, but could be toxic to other bacteria. So for a long time, so about 150 years ago, when germ theory, we'll talk about germ theory in the next unit, and when germs were first discovered, people were very slow to adopt them and very, very slow to adopt this idea that there was even such a thing as a germ or a microbe that could cause harm to us. But once it was accepted, it very quickly became the norm or the standard to say that any bacteria is a bad bacteria and everything has to be sterile and we should get rid of all bacteria at all costs. And only very recently have we kind of come around to this idea that we live in harmony with bacteria. And of course, that's called our microbiome. And there's a lot of really cool research being done on this. And some initial indicators of why bacteria are helpful to us came from these states of what we call dysbiosis. So for example, if someone is in the hospital and they're on a very, very heavy antibiotic regimen because of some type of infection, it's going to get rid of all the bacteria that's in their gut. And that's not good, because what tends to happen when all the bacteria go is it makes room for bad bacteria. And the perfect example of this is C. diff, Clostridium difficile. And Clostridium difficile can cause massive diarrhea to the point where you become dehydrated. And most people that get C. diff as a consequence of antibiotic use that are in the hospital die. It's a very, very dangerous state to be in, and it can be controlled by having normal microbiota in your gut. Another example of this is a yeast infection, Candida albicans. So if you're taking antibiotics, it's going to get rid of the normal microbiota in your body. For example, we have a lot of lactobacilli and a lot of lactobacilli, sorry, in our vagina, for example. And if you take antibiotics, and so those bacteria tend to cause a slightly acidic environment, I want to say. And when we get rid of those bacteria, the pH slightly changes, and now it becomes very hospitable for this yeast Candida albicans, and you can get a yeast infection. So that's one kind of very common outcome of taking antibiotics. And again, when your immune system is compromised, those different types of yeast can take over your body and can cause really bad gut problems and upper respiratory problems as well. The last one, as a Staph aureus, we should not have Staph aureus really on our body, and it's usually outcompeted for space by Staph epidermidis or Staph. What's another common staph species? There's several common staph species that are found on our skin, and we shouldn't see Staph aureus. And for some reason, in people with atopic dermatitis or eczema or psoriasis, what we see if we take a sample of the bacteria that are on their skin, the dominant bacteria on their skin is Staph aureus. It's not Staph epidermidis and other types of Cutibacteriums and Corynebacteria that are normally found there. So all of these these clinical cases led to this idea that bacteria are actually there to help us, and that there are a lot of bacteria that are part of our homeostasis maintenance, and they need to be there. All right, so now let's move on to our sensor system and imagine that we have an infection that has gotten past that first line of defense, so past our physical barriers and past some of those secretions that we have to protect us from external pathogens. But before we do that, I want to talk a little bit about the cells and structure of our immune system and how they work. So remember that we have stem cells in our body. Right now we have a bunch of hematopoietic stem cells, HSCs, that are found in our bone marrow. This is one of the reasons, kind of a less commonly thought about reason for the danger of catastrophic injury or amputation in our legs. If you get a crushing injury to your femur, or you lose your femur, or you get cancer where you have to have chemotherapy, you losing that bone marrow, you're basically losing your hematopoietic stem cells, which is devastating for your blood and your immune system, because that hematopoietic stem cell turns into all the cells of your immune system. So when you hear about someone undergoing chemotherapy, getting basically radiation treatment, and then getting a bone marrow transplant, this is what they're transplanting, are these hematopoietic stem cells, so that they can regenerate their own components of the blood and the immune system. So these hematopoietic stem cells get very specific signals. Think about it. We all started out as a zygote. We all started out as one cell. That one cell became two became four, and so on. And eventually we had, during the fetal development, we had these hematopoietic stem cells differentiate from the precursor stem cell. So this hematopoietic stem cell is going to get very specific signals that are going to turn this hematopoietic stem cell into two major types of cells, either a common myeloid progenitor cell or a common lymphoid progenitor cell. So we have two major kind of branches of our cellular components of our immune system and blood: myeloid cells and lymphoid cells. So let's look at the myeloid cells first. We're really not going to talk about this side. Let's see here. There we go. This side of development, because even though they're super important, right? We need our red blood cells and our platelets. They don't play any type of major role in our immune system. So we're going to pretty much ignore these. But what I do want to talk about is this and this one. So our granulocytes and our monocytes, so granulocytes here and monocytes here, are major players in our innate immune system. So our granulocytes can be categorized into three types of cells: eosinophils, basophils, and neutrophils. Whereas our monocytes can differentiate into two major effector cells of the innate immune system, and that's macrophages and dendritic cells. So when we talk about our leukocytes, we're talking about all of these. So anything that's in a green box right now is considered a leukocyte or white blood cell. But notice that we have three major kind of categories. We have granulocytes. So we have granulocytes, monocytes, and lymphocytes. So those are our three major categories of white blood cells. So when you do like a blood cell count, a WBC, if you look at someone's blood profile and you want to see what types of cells are present in your blood and at what levels, that's a good indicator of whether or not there's an active infection or there's something going on in your blood, because we know what the normal ranges are supposed to be, and so we can see if any of these look out of the ordinary. So for this particular lecture, we're going to focus on granulocytes and monocytes. Even though these are important, we're going to talk about them in the adaptive immune lecture. All right, so let's talk briefly about granulocytes first and just kind of characterize the three major granulocytes. Neutrophils. Neutrophils are the... Oh goodness, how would you even explain? Neutrophils are oh goodness, I don't even know how to describe them. They're just the all out crazy, like so self-destructive. They are the warriors that will just go nuts on the battlefield and kill everything, and they go so hard that they actually usually die within a day or two of becoming activated. And they're really one of the more important in terms of effector cells. So those cells that actually will have an effector function, they're probably the most important effector cell, because they are going to kill everything that they come into contact with, because they dump these really, really toxic chemicals and substances into their immediate surrounding environment. When they're recruited to the site of infection, they're just going to start releasing all of these enzymes and these different antimicrobial substances, and then they die. And in fact, if you've ever had a bad enough infection where you have pus, the pus is actually dead neutrophils. That's kind of gross to think about, but when you think about that white fluid that kind of accumulates at the site of infection, most of it is neutrophils that have just killed everything around it and killed themselves, basically, because they go hard. They're really hardcore cells of our innate immune system. But again, remember, this is very general. They basically get activated, they get recruited to the site of infection, and then they just start throwing grenades and do it until they run out of energy and then they die. On the other hand, we have basophils. They're not quite as hardcore as neutrophils, but the basophils' major role is to activate inflammation. And in fact, they activate inflammatory signals that recruit neutrophils. So they tend to be the cells, along with mast cells, that are most implicated in allergic reactions, because they secrete these little tiny chemicals called histamines. And histamines cause inflammation. So I'm sure if any of you have had allergies, this makes sense to you, because the medicine that you take to combat allergies, of course, are called antihistamines, because they're chemicals that neutralize the effect of histamines. So histamines are secreted by these basophils. It causes inflammation, vasodilation, and recruitment of cells like neutrophils, macrophages, and dendritic cells. So the third type of granulocyte is called an eosinophil. And eosinophils are, let me see what time it is. Okay. Eosinophils they're kind of, this isn't completely true. Again, nothing I'm saying in any of these lectures on the immune system are completely true, because again, our immune system is so complicated that there's a much larger and much more nuanced picture than what I'm telling you. But again, the stuff that I'm saying is generally true. So generally speaking, eosinophils are kind of an evolutionary holdover. They don't serve a lot of function right in our bodies now, because we are very fortunate to be living in an age where we don't really have parasites. If you think about even go back 3 or 4 generations, like our great grandparents, they probably had parasites, because of just the conditions that most humans were living in at that time. It was very likely that they had some type of multicellular parasite that was invading their body at any given time. of course, advances in public health and hygiene and so on have largely eliminated that possibility and have made eosinophils kind of not as necessary branch of our innate immune system. But their primary response was usually towards these multicellular invaders, for like tapeworms and nematodes and all those fun things that we don't have to think about anymore. All right, so continuing on with the different types of cells, let's move on to our monocytes. Notice that we can also call those mononuclear phagocytes. And remember, whenever you see this word phagocyte, you want to think about eating, right? Phaye, Greek word for eat. And so they're eating cells. And so the two cells that I want to talk about, of course, are macrophages and dendritic cells. And again, these are our sentinels. They're the ones that are going to patrol the perimeter and keep be ever vigilant and look for those foreign invaders. And macrophages in particular are found pretty much everywhere in our body. We have macrophages that are roaming throughout our blood and our lymph, but we also have very specialized macrophages that are found in specific cells or specific tissues of our body. For example, we have macrophages in our liver. We have macrophages in our lungs, in our abdominal cavity. We even have them in our brain, believe it or not. So these are all very important. So they're in these different areas of our body so that they're one of the first responders to infection. And once they phagocytize whatever pathogen that might be, they're going to migrate to our secondary lymphoid organs and alert the rest of our body that we have a pathogen present. And then the thing that I want to kind of point out about our dendritic cells, one, they look really cool. They kind of look like dendrites. That's what I think that's why they call them dendritic cells. They look like the dendritic what is that called? The dendritic process, or the dendritic part of our neuronal cells, right? Kind of highly branched and have all these kind of outward migrations from its center. And this is because it wants a lot of surface material. And this is because dendritic cells will interact with B cells and T cells in the adaptive immune response. So even though I'm including dendritic cells in the innate immune system, it's really a bridge to our adaptive immune system as well. So dendritic cells play a really big role in the adaptive immune system. So much so really that I wouldn't even say that dendritic cells are part of I wouldn't assign it. If it were up to me, I wouldn't necessarily assign them to the innate immune system. I would put them in the adaptive immune system, simply because they play such a huge role in the adaptive immune system. The reason that many people put them in the innate immune system is because they're nonspecific. When they start phagocytized, they get recruited to a point of contact for pathogen activity, and then they just start ingesting things. So it's not a specific response. All right, so I think I'm going to stop there. I'm almost at 50 minutes. I'm going to break this talk into two parts just because I'm talking way too much.